Okay, so I have many less data than all the previous speakers to show. So I will just introduce you and do a progress report of where we are with this project. That is the ancillary study of the PULSE cohort focusing on whole genome sequencing. So the reason of the rationale of the PULSE cohort has been quite nicely introduced in particular by Philippe, that ALS is very heterogeneous, and one of the reasons for that is that ALS is clinically diagnosed. So it relies on the double involvement of upper and lower motor neurons, and it's more probably, and we also had a philosophical discussion about that, it's more probably a syndrome than a single nosological entity. And there comes the problem. And the problem is amplified by at least two levels of heterogeneity. The first one is the heterogeneity of the clinical picture of ALS patients. So ALS patients can have predominant UMN involvement, like in PLS, predominant LMN involvement, like in PMA. They could have spinal onset, bulbar onset, and they could have a specific phenotype like flame arm syndrome. So very different clinical presentations that complicates the picture. A second level of heterogeneity is also the heterogeneity of the progression, which is actually related to the heterogeneity of the clinical picture. And here, these are ALS-FRS from 30 ALS patients from K11, and you can see that these ALS-FRS, they vary in slope quite widely, and this is something that is observed in many clinics, that some patients progress very fast within a few months, while some others can be maintained for many years. So the question from that is, how should we tackle this ALS heterogeneity? And we, surprisingly, in the field, lack a natural history cohort with multimodal assessment. There are not so many of these, and especially not prospectively and with associated biobank. So clearly, we need to characterize the heterogeneity prospectively, and we need to associate the clinical characterization to a biobank with longitudinal samples to enable a number of ancillary studies. And this is the PULSE cohort that has been initiated and highly supported by ARSLA. So the PULSE cohort is headed by David and Anne-Sophie, which are just there. And the whole French ALS community is actually involved into this cohort. There has been 500 patients included in ALS Reference Center. And you have heard one of the first ancillary studies that has been published by Pierre-François on MRI data just the session before. So it's a prospective, observational, multiparametric, and longitudinal study with recording of a number of clinical data, obviously demography, function, muscle testing, EMG, cognition, even polysomnography, and treatments. And importantly, biobanking of DNA, serum, CSF, etc., and imaging. And the objective goal, the primary endpoint of this is to identify the predictive factors of ALS survival. Okay, so this is PULSE as it was initially planned, but initially in this natural history cohort, there was little genetic to be included. And we thought in the community that it was probably something that needed to be solved. Why? Because among 500 patients, there are probably 50 C9, 10%. And C9 patients, they have more frequent cognitive impairment. You can see it here from this paper from Michael Benatar, where the ECAS score of C9 positive patients declines with the progression of the disease. So if we want to study endophenotypes, we need to know who is C9 and who is not C9. We need to know who is TBK1, who is not TBK1 for the same reason. C9 are 72 patients, they also progress faster. So we need to identify these. SOD1, we need to identify the mutations because we know that there are mutations that are fast and we know that there are mutations that are slow. First patients are also, so probably between 5 and 10 of the PULSE cohort are likely to be first mutated, and they are more frequent, they have more frequent aggressive disease with younger onset. So clearly, one of the aims of the genetics is to identify the familial monogenic causes in the patients already included in the PULSE cohort. But that goes way beyond that because I think we also need to identify the common variants. Why? Because there are common risk factors which are present in the population very widely, in a much more frequent manner than, for instance, C9, obviously, that are known genetic modifiers of ALS. And one of these is obviously UNC13A. So UNC13A, we are, I don't know, 200 in the room, probably 40 of us carry the risk allele. So among the 500 cases included in the PULSE cohort, there are many homozygous or heterozygous carriers of the UNC13A risk allele, and we need to identify those to be able to really predict the factors that predict survival of ALS. This is just one of the many studies that showed that UNC13A genotype, here the CC allele that Philippe alluded to, these patients, they have faster disease progression than the ones that do not carry the disease, the risk factor. So adding a genetic branch to the PULSE cohort is really invaluable in terms of interpretation of what we will know, what we will learn from this cohort. So this is what we did with Lille and with 3 other labs. We are doing the whole genome sequencing and are responsible for this in Strasbourg. And the lab of Serge Limbroso and Claire Guissard is doing C9 gene scan in Nîmes, and Patrick Vourch and Philippe Corsia are doing the C9 scan in Tours. So half of the patients will be determined for this in each center. So the reason why we do C9orf72 gene scan in addition of whole genome sequencing is because we miss most of the repeat expansions with the whole genome sequencing. At least we will be sure of this C9 repeat expansion with this second endpoint. So what will we do? So first, we have 4 outcomes. First, performing and obtaining the sequences. Second, the C9 gene scan. Then we will identify all the ALS-related gene variants, and we will call for all the known genes panel as a panel into these 2 data sets. And fourth, and more prospectively, we aim at systematically correlating genetics with progression and endophenotype, and I will explain to you how we will do that. So where are we now? This is ongoing work. It's been 2 very tough years, mostly from the administrative point of view, and I guess David and I lost our last piece of air within these 2 years because of these GDPR constraints that are linked to this genomic data, and that are now very rigorously let's say, applied by our institutions. So the biological samples have been sent to center, and the C9orf72 gene scan is almost done. With respect to the whole genome sequencing, more than half of the sequences have been done. And the analytical pipeline are up and running. So as early as we got the data, we can begin to really align these and call for the variants. Just one note of caution. This is a very, very computational demanding project. More than 6 months of computation are actually needed in our data center to really do properly the work. So on the short term, the whole genome sequencing will be finished within 24, and we hope for spring 25 to have aligned and called the variants and be able to have rigorous identification of the pathogenic variants present in the cohort. We will do a primary analysis of the known pathogenic, but also the variants of unknown significance in known ALS genes. And the aim of the Spring 25 will be to submit a paper on the clinical genetic analysis of the cohort. I think there are not so many papers that relate genetics to clinics on a natural history cohort like this. And last but not least, in parallel, we want to grant access to the whole community to these data sets. So we are building up what is called in French an entrepôt de données de santé. In English, it's a data warehouse that will enable us to grant access to the community to this data under the umbrella of our SAB that is headed by Claude Denuel. So that's the goal of the next, hopefully, year. In the long run, what we want to do is to integrate I will just say one example of what we will do in my lab. But if you have ideas, the data will be available for the community. We want to integrate PULSE whole genome sequencing with other cohorts to meta-analyze genes of disease progression. So right now in my lab, we've obtained data from 2,300 patients with longitudinal ALS-FRS, which are QC'd, and for which we have, for 2/3 of them, whole genome sequencing, and the rest is high-density genotyping. And we already have some very interesting hits in these GWAS of ALS-FRS. So the PULSE cohort is a very nice replication cohort for this, and will be integrated in a meta-analysis. On a more general scale, this is a cohort that will, without doubt, be very useful for understanding the genotype-phenotype correlations, for instance, weight loss, for instance, cognitive impairment. Are there specific genetic factors which are related to weight loss or cognitive impairment? This is a cohort we can explore for this, even if we need to have a number of other cohorts to meta-analyze and probably reach statistical power. So this is a work that is headed in my lab by someone you've already heard of in the talk of Caroline, and I promise that I will push him to be there next year, because so many people told me, where is Salim? He's in Strasbourg right now. And I want to acknowledge, obviously, the PULSE team, and you can see here David with a pipette, probably is already pipetting the DNAs to send to the platform. And Sophie, which are the two people that led this project and made it alive. And obviously I want to thank all the participating centers, the SAB of PULSE and the centers of NIM and ARSLA, and I realize that I do not have the right logo, but all my apologies, but thank you for the support.
Thank you. So, any questions, comments? David, you want to make comments?
Yes. I apologize, everyone. Well, we are losing time, but then after we will gain time, because it will be open for each one of you. For any idea, then we can mix genomics and proteomics, etc. So we are trying to set up this health data hub. So it's a huge work. So the genetic data will be held in Strasbourg, and it will be also with France 2030. So we are going toward a large possibility of exchange for every researcher in the world. Of course, that's what ARSLA want and what we do need to do.
Vas-y, je vois.
Very nice initiative, and especially the link with the detailed clinical phenotyping and biosamples. You know, in Project Mine, we maybe don't have the large clinical data set, but huge sample size. And even there, we struggle with all the rare variants. So how will you tackle and categorize the variants of unknown significance? Because
that's a question for my fellow geneticist to answer. That's a good question. So first we will identify the known pathogenic variants. Then we will identify the possible pathogenic and the views in the known, just like everyone, I guess. And then if we want to be sure that these patients have actually pathogenic, then we will have to turn to functional studies. But maybe Kevin or Claire can comment on that.
I think the relationship between both could be interesting as well.
Yeah.
Because mine can help
The variant
one, and then we can go back to PULSE to have more detailed analysis.
I did not answer on mine. So mine is an invaluable data set. We are in contact with Jan because we are comparing the GWAS. So he's doing the GWAS of survival, and we are doing ALS-FRS. And actually, we are popping up UNC13A in our. So that's quite reassuring that known things are popping up on both sides. I think the survival needs really 2, 3, 4 orders of magnitude more patients because of the noise. Individual decision, national legislations are really. So this is difficult, both on the diagnosis and both on the death part. So probably the fact that there's less signal in the survival is due to this, but we will compare and we will exchange with mine, obviously. For the rare variants, we are doing burden analysis also. So.
Thank you for your insights in this great initiative. I have a comment. We run in Germany a multi-center biomarker study, including 4K patients now, and we also do phenotyping in a pretty harmonized way. And the question is whether it would make sense to align, or at least to exchange how we do actually phenotyping. So that also involves the propagation phenotypes such as flare leg, flare arm, and things like that. So you do that, we do that, but I'm sure we don't do that the same way.
Sure.
And it would be of interest to have an exchange on that.
Yeah. And are you doing also genomics?
No. So
do you have DNA?
Yeah, I mean, we do genomics, but not to that extent, just for genes. And,
okay, but so you have DNA?
So we yeah, but not in 2K, not in 4K, but only in 2K patients, right?
Oh, cool. Perfect.